Welcome to Barry's In the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Welcome, everybody, back to In the Interim. Today, we're actually going to take you in the interim. We're going to take you into many interims. A unique look back at a trial, a drug, a disease. And I have a wonderful guest. Don Barry is here, my father, to join me. And we're going to talk a little bit about the story of BAN 2401 and the story of a phase 2 trial and the before and the after. So welcome back to In the Interim. So let's go back to approximately 2013. And the sorry state of Alzheimer's disease, drug development, something like 14 straight failed phase 3 trials, promise of drugs, but all of these trials failing. And take us back to that time and set the stage a little bit, Don.
Okay. It was, as you indicate, a sorry time, but Alzheimer's researchers, to their credit, understood that they were doing something wrong. They didn't know what. And the Alzheimer's Association put together a roundtable, a meeting in Washington, which had the goal to study and learn from, it was a bit of a workshop, the 14 latest phase 3 trials in Alzheimer's, all of which failed. And so there was each of the trialists were asked to present what the trial was, and to the extent that they could, why it failed. Was it the drug? Was it the trial? And they had invited me to give a presentation. I mean, they were aware of I-SPY 2, which was a trial in cancer, but did smart things along the way. And so they were, like many researchers at the time, sort of salivating over doing something like I-SPY 2. So I presented my analyses of what the problem was. One of the, I won't go through all 14, of course, but one I want to mention is a large pharma company that had a drug that they were looking at for Alzheimer's, and they had a phase 2 trial, and it led to a large phase 3 trial that bombed completely. The person presenting was one of the instrumental people in the company who was part of the design of the phase 2 trial, was part of the analysis to go forward into phase 3, and he said explicitly, we didn't know that we had a good drug here. We recognized that we might fail. The data were not all that consuming. Of course, what the down part was in the cognition and function, and the drug was getting in, it was doing what it was supposed to be doing, and the biomarkers that they could measure, but it was not big enough, so to speak, to show cognition and function. And so it was an admission, and the person was asked, okay, so why did you do the trial? Why did you do the phase 3 trial if you didn't have enough evidence that the drug was going to have an effect. And he essentially said, well, yes, we were playing the lottery. That is, we're taking a chance, and, the chance might be based on a phase 3 trial that just randomly, 5% of the time, even though you don't have anything, you have a positive trial. So he was I thought surprising, and this happens a lot, that you don't know, that you sort of hope for the best. So we're in World Series mode, so we have a pitcher that's a great pitcher, and you're swinging for the fences, and you're hoping to both hit the ball and hit it right and hit it into the fences. So there's a lot of randomness. Let's hope for the best. The result of that was, at the meeting, was Veronica Logovinsky, who was at the time at ASI, and she asked if she could discuss with me their circumstance with the ban 2401. So I agreed, and I visited them, and they were enthusiastic, and that's really what it takes, is enthusiasm, part of the company. They were enthusiastic about doing something different. They ended up doing something different. And Scott, back to you.
Okay, so sorry state of drug development, question of whether it's the drugs, whether the trial designs, whether it's the development plan, whether it's actually patients and endpoints. Do these patients actually have Alzheimer's disease? Are we measuring the right endpoints? Lots of blame going around. So as you mentioned, ASI and Veronica Logovinsky, Andy Satlin, Chad Swanson, Shobha Dada, Jinping also was involved in this, Jinping Wang. So a large number of people, large effort. We built a phase 2 trial. And the notion was we want a better phase 2 trial. We don't want to just jump into a phase 3 trial. So I'll describe the trial somewhat briefly, but the trial design had 5 different arms of band 2401, the drug. It had 2 different frequencies. The bi-monthly frequency had 3 doses, and the once-monthly frequency had 2 doses. So 5 active arms and 1 placebo arm. In order to incorporate the 5 arms and not have a much larger trial, response adaptive randomization was used in the trial. So interim analyses would take place, and allocation would be focused on the arms that were doing better. The endpoints in the trial sort of plays an interesting role here, 12 months and 18 months, change from baseline in a cognitive measure in this patient population called ADCOMS. ADCOMS is a collection of questions from MMSE, ADAS-Cog, and CDR sum of boxes that was thought to be highly sensitive in this part of the disease. And so this was the endpoint in the trial. In order to drive the adaptations, the RAR, we built a model, a longitudinal model that would look at 3 months, 6 months, 9 months, 12 months, 15 and 18, and forecasting forward where the patient was to identify within patient that forecast, to identify how well the arms are working and to drive the adaptation. So a key part of this is what we'll call the longitudinal model within that. Adaptive analyses, the trial could stop for futility. They wanted to be realistic about this, given the sorry state of Alzheimer's development. They also wanted triggers for accelerating drug development. So if a particular trigger was hit, they would go quickly to phase 3. If it wasn't futile and it wasn't this trigger of phase 3, it would go to the next interim analysis, and it would get more data and more data during the course of this. So this is largely the trial design within it. Now, the sample size also was adaptive. 300 was the minimum, and then 800 was the maximum. If we didn't know the answer yet and we kept going, we'd enroll to 800. We would then continue interim analyses as we're getting exposure through 18 months, the trial design. Now, this is a unique trial in terms of disclosure. Eisai has published the result of every one of the interim analyses during the course of this trial, and there were 16 of them during the course of this trial. So many interim analyses during the course of this trial. And I think we'll take you through the story of the trial, what happened, and the going forward. But any other comments about the design before we sort of walk through the trial and what happened, Don?
So the interesting thing is what happened in the trial. No, I think you just get into that.
Yeah. Let's. I know where you're going, and let's not quite go there yet. Within it, okay. So all of this has been disclosed. Interestingly, in the course of this, is that in the trial design, I was unblinded and part of a group that was reviewing the interim analyses. Don was blinded. Don did not see the data. He was involved in the design. We were both involved in the design, but he was blinded to any results going on during the course of the trial. The trial was conducted by a company called Tesela, that they ran the interim analyses, they curated the data, ran the interim analyses, and then I was involved in reviewing them with Christine Brolio, who's also here at Barry Consultants. So the first interim analysis in the trial happens, 196 patients enrolled. It happens in the date of this, I don't want to get this wrong, the date of this is March 2nd, 2014. So the trial design, as Don described, happened in the time of this. And at that time, randomization is updated for each of the arms in the trial. Even at this first interim analysis, the randomization, and this is published with this, and we'll add a link to this, is randomization to the 2 higher doses is increased, and each of the other 3 doses is decreased. The placebo is held constant. The trial does not hit futility. It can't hit success at this time or flagging to Eisai to go to phase 3. So the trial continues on. One comment about flagging to Eisai is they were interested if there was a high posterior probability that their effect was at least a 25% slowing. That was kind of this important trigger for them. If they were highly likely of a 25% slowing, they wanted to know that, be told that, and they would accelerate to phase 3. If there was a low probability of a 25% slowing, they would stop for futility. Okay, that continues on every 50 patients. So at 250 and 300, similar analyses go on. Now at 300 is where an unplanned event happens in the trial.
So can I interrupt you and say a little bit more about
Yep.
So the 300, when we talk about these things, a lot of people say, oh, well, you need, of course, complete data. This is not complete data. This is all the patients in the trial. Nobody is at the 12-month point at this time, the first analysis. But we're using the longitudinal model to predict what's going to happen in the future. So, and the most interesting thing that happened at this first analysis was that the algorithm was already learning that the two lowest doses were not effective. And so it essentially zeroed out. It wasn't quite zero the first time, the adaptive randomization. And it was built so that they could come back if the longer term data pointed out that, you're missing something, and now the low doses are actually doing well. That never happened. Something to address, Scott, and something that the journalists addressed and quoted people in the research community was the many interims. I mean, I was just taking it as a negative. You're doing all these things and you're going to be misled. And there was lots of criticism about simply the multitude of interim analyses. Would you comment on that?
Yeah, and we'll come back to this in part of the story. And it's general, and now there's this pervasive viewpoint that it's too complicated, these number of interim analyses. But every 50 patients, an analysis was done. And a key part of the design is we're only stopping at that point if we have strong evidence, a 95% chance that we have at least a 25% slowing, or less than a 7.5% chance that we have a 25% slowing. So, but if we're not there, we're going to continue on in the trial. So, and then we're going to update the randomization as Don described in that. So this trial and its ability to update was critical. And the analyses, every 50 patients enrolled, was critical, as we'll describe what happened as the trial goes.
So, as Scott mentioned, the fact that these are available, the interim analyses, well, one of the places is a publication that I was first author and Scott was last author in JAMA Network Open. And what you should do, dear viewers, is go to the supplemental material, and you can actually watch a movie. You can show a movie of what happened in all of these analyses and how did things change. Naturally, there's correlation from one to the next, but you're looking at the tendency of where it's going, what it's doing. It's assigning to the higher doses and control along with it. Control had the same probability as the highest probability experimental drug. And it's very instructive. It's not, these are obviously not independent. We're taking these serial looks, and it's one of the few studies that all of the interim analyses are published in. Back to you, Scott.
Yeah. So the first interim analysis, and Don described that it's triggered by patients enrolled. And I'll back up. There's two Bayesian models that are critical to this. One is a dose response. So the 12-month change from baseline in ADAS-Cog is modeled in a two-dimensional normal dynamic linear model over the 5 arms and placebo. The other one that turns out to be even more critical is we built a model between each visit and 12 months for the correlation between them, built a linear model between each one of these, that a patient's change from baseline at 3, 6, 9, wherever their last exposure was, is used to estimate what is their value going to be at 12 months. Multiply imputed through the model to estimate the dose response and drive conclusions. So as you're flipping through this, as Don describes, at 300 patients, it's a bit of a fun movie to watch. At 300 patients, the data's at its worst relative the arms to placebo. The placebo is actually at that point estimated with high uncertainty to be the best arm. And the probability that the 10 bi-monthly dose is better than placebo, is as a 25% improvement over placebo is 18%. Had that have dropped below 5%, the trial would have stopped for futility. And that's the closest it's ever got to futility. It moves on to 350. So now, by the way, we are in the September 26th of 2014. So we've moved on about 7 months. They've enrolled 250 patients. They're on their 4th interim analysis. And enrollment is temporarily paused in the trial. There's a safety committee that's looking at the data, and they notice a radiological finding. We now understand this in the disease as ARIA, and it is a radiological finding that looks like microbleeds in the brain, and specifically for the highest magnitude dose, which is 10 bi-monthly, they are noticing this for patients that are ApoE4 positive. They are asymptomatic. So these are not symptomatic bleeds in the brain. They're asymptomatic, but they were concerned about what is this, to the extent that they said ASI could not enroll that high dose. One of the 5 arms could not enroll patients ApoE4 positive on that arm. So we've built this adaptive randomization for all of the different arms. One of the stratification factors is ApoE4 positive. At that time, the decision is made by people blinded in the trial that we're just going to separate that randomization for ApoE4 positives and ApoE4 negatives. And there'll be different randomizations, adaptive randomizations, which will zero out the 10 bi-monthly. And the trial continues at that point.
The patients who had been assigned to that
Right.
within the last period of time would be stopped. So it was, it was like an A-bomb dropping on the trial. And ASI was very concerned. They knew, of course, about this. They got the message from the regulators, and they had to do it. And they talked to me about, canceling the trial. It was such a huge negative. Scott?
So this is one of those cases where an unplanned adaptation was able to very quickly, by the way, Tisela did a fabulous job, that now it's randomizing patients with this separated randomization for them. And the trial continues on. At this point, by the way, at 350 patients in, it wants to randomize. It has absolutely zeroed out the 3 low doses. The 2.5, the 5 monthly, and the 5 bi-monthly. It wants the 2 high doses. So ApoE4 negatives are getting the 10 bi-monthly at a high rate. The positives are getting only the 10 monthly at that point. And it is already focused at this point in the trial on the high doses. And now the estimated efficacy, the probability of at least a 25% slowing, is about 55%. Again, if that's 95, they would have signaled to ASI back in 2014 to run phase 3, but it did not signal. It says, the data's good, but keep going. Its estimated efficacy at that point is approximately a 25% slowing. This continues on, and interestingly, and so now at this point, only about 15 patients per dose have passed 12 months. And it's estimating that high dose has about a 25% slowing through this longitudinal modeling. The early patients enrolled are starting to move past 12 months.
And data, and the Scott Berry's model for missing data.
Yeah, so on the 10 bi-monthly dose, ApoE4 positives that had less than 6 months exposure were removed entirely. So there were different kinds of missing data. There were patients in the trial who went away. There were patients that were forced to go off drug. And then there were patients that were enrolled so frequently that we just didn't have their 12-month data. All three of these types were being imputed based on their last cognitive status in the multiple imputation model. 500 patients enrolled, so this is June 19th. By the way, these interims took 2 or 3 days to run. The data was there, the algorithms were run. That largely took a day. They were reviewed in 12 hours a day. They were signed that they were good to go, and they were implemented immediately. The information was passed to a data safety monitoring board, but these were done essentially fully automated with a review to make sure nothing weird was going on. And the trial continued. At this point, it's estimating about a 25% slowing, which happens to be in this really interesting part for ASI. If there's high probability it's better than 25, they want to run phase 3. If there's low probability, they would want to stop for futility. And it's hovering around that point.
And that's on the basis of the imputation of the missing data. Had you looked at just the raw data and ignored this, you would have had something like, 10% or something a lot less than 25%.
Yep.
And the DSMB, were they happy with the model, or did they react to the model? Because everything's driven by this model.
Yes, they had no concerns with the model. In it, we actually created, and we only interacted with them infrequently. The interims took place. We said statistically they're good, and they signed off on it and continued. It was this one aspect of the ApoE4 positives, and that continued throughout the trial. This continues on at every one of these interim analyses, and so the trial gets to its full enrollment. It actually over-enrolled 856 patients, ended up over-enrolling in the trial. And at this point in the trial, this takes place, full enrollment takes place in September of 2016. All patients are enrolled. At this point, the only thing, it can stop for futility, and it never got close to futility again after that third interim, or it could signal to ASI. Now the ASI built in a really cool part of this trial, that this all is being built, is being triggered on 12 months change, but all patients are being followed through 18 months. And the trial's going to run and get full exposure through 18 months. And at 12 months, this was billed publicly as, we'll find out if this is going to go to phase 3. And at this point, they have that if there's an 80%, they lowered the threshold, if there's an 80% probability of a 25% slowing, they wanted to go to phase 3 in that. And again, they could stop for futility. They found out at this point it's not 80%. And the actual value, let me find out what the actual value of this was at the time that this interim analysis happened. At the time this interim analysis happens, when all patients are enrolled, the probability of at least a 25% slowing was 65%. So that needed to be 80, and it was 65%. So it didn't jump that 80. So ASI publicly disclosed this, that we're continuing exposure in the trial. We don't know the answer yet. And interestingly, I'll go to a snapshot of an article that was written by in the Alzheimer's forum about this result. And they interviewed you, Don, and asked you about this. And they also interviewed a neurologist, a well-known Alzheimer's trialist, Paul Azen. So they asked you about this result that at 12 months, we don't know the answer yet, and Don is blinded. And he says the band 2401 trial results so far, to the extent that we know them, and all he knew was the trial was continuing, it wasn't 80% and it wasn't less than 7.5%, indicate the therapy is neither extremely effective nor clearly ineffective. On balance, and in context of the sorry state of drug development in Alzheimer's disease, that's good news, was Don's quote at this. And we are now 6 months from full readout of the trial. And all they know is it's not 80 and it's not less than 7.5. Paul Azen takes an opposite view, and I'll read you his quote, and I think it's kind of interesting. So what do these results mean for the field? Not everyone is convinced that adaptive trials are the wave of the future. Paul Azen of the University of South California in San Diego commented that while adaptive designs have proven useful in the oncology field, where endpoints are more objective, they may be problematic in AD trials, Alzheimer's disease trials. And now quote, the greatest concern about Bayesian adaptive approaches in AD trials relates to our cognitive clinical outcome measures. These measures are highly variable and noisy. Adapting on interim, incomplete data risks chasing noise and making erroneous decisions regarding efficacy or futility.
So I'm just compelled to say, I know that it has noise. I know this variability. I recognize it. I modeled it. I live with it. And I can tell and build into the algorithms, in the design, what's real and what's noise.
And what was so misunderstood by Paul is that what did the adaptive design say? It said, we don't know the answer yet. Let's keep going. We need more data. We don't know the answer. And his criticism was they're too risky of making erroneous decision, claiming success or futility, when in fact it knew that the variability, and it knew keep going, go to 18 months in it. So there was this sort of doubt, and it was built in and part of it. Okay, flash forward now 6 months, and the trial finally reads out 18 months. And by the way, the randomization, so the adaptive randomization, what's the role of this adaptive randomization? There, out of the 856 patients enrolled in the trial, 238 go to placebo. The lowest dose gets 52 patients. The next gets 48. The other 5 milligram gets 89. Those are the 3 doses that very quickly are moved away from. The 2 high doses get 246 and 152. And the only reason it was 152 is because it couldn't enroll ApoE4 positives. But at that time, out of the band 2401 doses, 587 get put on band 2401. 2/3 of them, 67%, 398, are on the 2 high doses. So the response adaptive randomization, when Don goes back to the Alzheimer's and says, you need better dose finding trials with better exposure to understand efficacy, you need to explore dose, it did that, and through response adaptive randomization, very quickly focuses on the high doses.
And so you get greater precision associated with the high doses, exactly the doses you're thinking about moving forward. So it's a huge benefit. But I want to point out that this is an attraction for patients. They can be told in the design that if there is a dose that is doing well, we're going to keep looking at that. And so you are, if that has happened in the trial, you're more likely to get a dose that's doing well. And not only in this trial, but in other trials that we use adaptive randomization, we find out that that's exactly true, that the end result is that the patients in the trial, this has been something that has been batted around for decades in JAMA, et cetera, is are patients in clinical trials treated better than patients not in clinical trials? And some say yes, some say no, and there's data on both sides. Here it's clearly the case that if you're in an adaptively randomized trial and there is something that's positive, you're more likely to get it. So it should be an attraction to not only physicians, but also their patients.
So the trial reads out at 18 months, and the two high doses that I described with the larger sample size, the 18-month estimate for the 10 monthly, so this is the high dose that's given monthly, and then there's the high dose that's given bi-monthly. The 10 monthly is estimated to be an 18% slowing. And its probability of being better than placebo is 93%. But it isn't estimated to be 25. The high dose given twice monthly is estimated to be a 27% slowing. And again, think about where this is relative to their targeted 25, and a 76% probability that it's at least a 25% slowing is the estimate of that dose. So ASI at this point does a couple things. It starts a phase 3 trial, and it selects the 10 bi-monthly. At this point, ARIA is much better understood, and there's actually some sense that this is a level of efficacy of the clearing of amyloid. So in the background, the other thing is the mechanism of this treatment is the removal of the pathological amyloid. And they're doing PET scanning, and the high dose is absolutely removing amyloid within this, and this ARIA is thought perhaps to be an effect of this, in some sense almost that it's showing benefit by this demonstration of ARIA. It's still a concern, and of course, if symptomatic bleeding or a higher level bleeding would, of course, be a bad thing within this. So they run this phase 3 trial, but they also submit the data to the agency, the phase 2 data to the agency, and present to them. So they plan a trial of the 10 bi-monthly, enrolling both ApoE4 positives and negatives, and a 1,795 patient trial is enrolled. Meanwhile, the FDA reviews the data. They give accelerated approval to BAN 2401, which has now changed its name to Lecanemab. They grant accelerated approval based on the reduction in amyloid. And there's a stunningly cool data that if you look at the reduction in amyloid for the lower doses relative to the higher doses, and you look at the change in the cognitive values for those doses, it's incredibly predictive. And I think the fact that the multiple doses and the effect on amyloid relative to the effect on the clinical outcome is very highly predicted by the effect on it, almost demonstrating, and I know we shouldn't say the word, a surrogacy of this, I think enabled them to trigger accelerated approval. Now the phase 3 trial reads out, 1,795 patients reads out, and the press release for this comes out in 2022, and it demonstrates highly statistically significant benefit on CDR sum of boxes for that dose at 18 months, and the estimated slowing of the rate of decline is 27%. Okay, it was nailed. Normally, to variability, we wouldn't have expected exactly, but it was exactly the estimate from the phase 2 trial in that Bayesian modeling that's doing the longitudinal modeling, that's doing the dose response, estimated it to be a 27% slowing at 18 months, and it hit it right on the head in that, which ended up to be highly statistically significant. And FDA gave Lecanemab full approval for the treatment of Alzheimer's disease.
Can I go back to that reading we had with the 14 failures in phase 2? It's hard not to wonder whether if they had done something in those 14 trials like this, saw something that was positive and increased the sample size accordingly, if we wouldn't have better treatments today for Alzheimer's disease.
Yeah, it's hard to go back to this trial, but I describe a couple things happened that I think was powerful. If you go back to the algorithms, and you can go back to this and look at the posterior estimate of them, when there are 500 patients enrolled in this trial, and this happens, 500 patients enrolled, this happens in 2014. The algorithm is estimating this 25% reduction, and it holds pretty constant all the way through the end, that it's giving this forecast. Now, I think Eisai did a great thing by getting this exposure and this decision, but it pretty much forecasted at that point. But I also alluded to back at 300 patients, when 300 were enrolled, this analysis took place for futility. The data were not promising. Had they run a 300-patient trial, a small phase 2, they might not have ever continued development of this drug. So the alternative designs that could have been run may have led to this never being run in phase 3. It's hard to know, and it's hard to know the decisions that would have been made in this scenario.
And also, had they used something other than a reasonable and somewhat sophisticated missing data imputation process, instead of that 27% that they observed in the phase 2 study, and they looked at just completers, it would have been 12%. In part, and this is partly possible to explain on the basis of the fact that the missing data were precisely, if you look at what the missing data were for patients who had been assigned to the high dose, that distribution was very different from the missing distribution for the other doses, and for example, for control. And so the model recognized that it was that whether the data is missing. Of course, the model didn't know about this regulatory folks that mandated dropping that dose from the future part of the trial. It only knew what the data were and was basing it on what the longitudinal modeling was. So it was, there are a number of, it's sort of a perfect storm, or not a storm at all, but a perfect weather, that everything was moving in the right direction, and it gave rise to a positive conclusion. What's the latest? I mean, Lecanemab has been fully approved. Now another phase 3 trial has been observed with similar mechanism of action. And can you say something about the current status, Scott?
Yeah, so donanemab from Eli Lilly has been approved, full approval. and it demonstrates slightly better slowing of cognition, I think, at 18 months. But now I think there are 2 treatments that are disease-modifying that have been approved. And interestingly now, where Alzheimer's was a bit of a dead end for pharmaceutical development, there's a huge investment now, in part that clearly that affecting amyloid has an effect, and it has an effect on slowing disease. Now, they're going into tau, going into better effects of this. There's much more in the way of development. I want to point out something interesting.
We always wanted to change the world.
Yeah. Yeah. And so the interesting thing is that trial design was run. Meanwhile, there were more than ASI that were interested in Don's messaging of better phase 2 trials. And Abbott was one of them. And Rob Lenz was the clinical in charge of neurology at Abbott at the time. And they ran 2 trials, one of them ABT-126, which is published. The results of that stopped for futility. And I think largely the conclusion was that the drug didn't work. So a very similar design run in that scenario with a different drug sort of was pretty strong that the drug doesn't work. And I think that's the right conclusion. So the design doesn't make the drug, but hopefully the design gets the right answer for the drug. And I think it's 2 examples of putting in a different profile to a very similar design gave very different answers at the end of the day.
There was an interesting discussion with Rob and the clinical trial committee at Abbott. They asked him at the beginning, so this is a complicated trial. I mean, it had many aspects very much in common with the 2401 trial. How much time did it take to do this? And Rob said, you're right, it took a while. It took us like 6 months working with the berries to get what we wanted. But on the other hand, the trial ended 2 years sooner than it would have otherwise because we were doing the right thing and investigating and understanding that we really did hit futility with that drug. So it's good in both directions.
Yeah. I guess a last thing it would be good to come back to is you made this comment. So in the trial, there were 17 analyses of this data. The data is accruing, it changed the randomization. But this notion that what's beautiful in the Bayesian perspective is that the analysis at the end of the trial is unaffected by the fact that all these analyses take place within it. And it makes so much sense. And there's this view that somehow these 17 analyses infect the data, that it no longer tells us with the same level of strength. And in viewing this and seeing the results, go through and look at this and just, we should be doing this in most of our phase 2 trials. It's good for patients. It's good for drug development. It's not a bad thing that these analyses take place. When a snapshot of the data are done, it doesn't change the data. It doesn't change the scientific conclusion. And I think this is a beautiful example of exactly that.
And in a way, it's AI. You know, everybody's talking about AI. But this is a prospectively designed trial. There's an algorithm that's run it. The algorithm is like an automaton. And so we can evaluate, as we did, the type 1 error associated with it, the power of the study, and the again, Tesselotone Park, working on the logistics associated with it. You know, we are hearkening back. Tom and I first worked together in a Pfizer trial where we had daily stroke trial, where we had daily updates, and the algorithm would re-randomize, would reassess the randomization probabilities every day. And this kind of thing, there is a logistical aspect of this, but it's possible to do, and it's possible to have something that's rigorous, and the FDA has issued guidance to address the question of the complex adaptive designs. So the FDA is wholly involved here, not only the neurological part of the cancer part, but other parts as well.
All right. So again, JAMA Network Open, Berry et al., Lecanemab for patients with early Alzheimer's disease, Bayesian analysis of a phase 2b dose-finding randomized clinical trial, has a really unique look at a trial that with a very cool story and a very cool impact. And so here on In the Interim, we've taken you through 17 interims and a look back at a pretty cool trial. So Don, thanks for joining me in the interim. I know you could not join me in the interim in that trial, but thanks for joining me here.
Thank you.
Till next time.